Hi everyone, welcome to our NMA presentation about decoding the activity in the superior colliculus during a bidirectional motor task. In our project, we decided to focus on the Steinmetz datasets, which contains the neuropixel recording of the activity of 30,000 neurons across 42 brain regions during a visual discrimination task performed by a mouse. The behavioral task in this dataset was a two alternative forced choice in which in each trial, a visual stimuli of varying contrast was shown either on the right, the left, on both sides, or on either side on the screens of the animal. If the animal performed the correct motion associated with the stimulus, they would get a water reward. So it was also designed with the go-no-go task. We decided to focus on the neuronal activity in the superior colliculus, the motor-related region, because this region integrates sensory motor information and receives input from the somatosensory cortex, the visual cortex, and also the primary and second motor area. Specifically, our question was to understand which brain region best predict the neuronal activity in the superior colliculus motor-related during the 2FC task. Let's jump to the method section.
So we use a Poisson generalized linear model approach to predict activity in SCM neurons, totaling 101 SCM neurons across 7 sessions. And the model inputs consisted of behavioral variables, including the motion of the wheel during the task, the face energy, and the pupil focus, where the pupil was focused on the screen. In addition, we use activity from all neurons in other brain regions that were recorded simultaneously with the SCM in these sessions. And we ran a PCA on all of this neuronal data and used the first principal component from each brain region as a variable in the model. Now, in a separate model, we used all of the activity from all neurons, but time shifted 50 milliseconds before the SCM neuron activity time point that we were predicting to look at the influence of time lag on predictability. And finally, we included a variable to encode trial type, i.e. ipsilateral versus contralateral correct trials. And we compared these models by using a base model only consisting of behavior to see how behavior alone can predict SCM activity. Then in the second model, we used the first principal component from the other brain regions recorded simultaneously. And finally, we added the variable encoding trial type. And crucially, in all of these models, we used a 5-fold cross-validation approach to split the data into training and test sets with 5 different folds. And because we didn't tune any hyperparameters, this was the only data splitting that we did. So this was the modeling approach, and now we can proceed to the results.
So here it shows the results by fitting different models. On the left, it shows the result from 2 example SCM neurons. We used explained variance as the metric to measure model performance. Here, the x-axis indicates the model type. The first one is the behavior. The second is behavior plus regions. And the third is the behavior plus regions plus model types. So the neuron on the left shows the best fitting by model type 3, which considered all the components. And each line here corresponds to 1 fold in the cross-validation process. The neuron on the right shows better fitting by adding other regions on top of behavior, but adding child type doesn't further improve the model performance. In the middle right, it shows population results of all the SCM neurons with firing rate larger than 5 Hz. So each line here corresponds to a single neuron, and the dividing plot is shown at the background. And so you can see some neurons has explained variance up to like 40%, which is pretty good. So we then group neurons, all the neurons based on the model that best predicted their activity. As you can see, the majority of the neurons are best predicted by model type 3, which considers all the components. And next, you can see how each component contributes to the model performance.
Yes, then we ask which factor would predict neural activity in superior colliculus motor region. So here we plotted the absolute beta values for each predictor region on a bar graph. You can see that, first of all, the behavior information such as wheel motion provide high predictability to the SCM activity. Furthermore, several visual-related regions such as lateral genetic nucleus and also the anterior medial visual regions show high predictability. The medial reticular nucleus also show good predictability on a time lag, which is 50 milliseconds time shifted. So in conclusion, our model on average explains 7% of variance in SCM firing. Both behavior results and activity in several visual regions and reticular nucleus show predictability to the activity of the SCM. These results indicate potential causal relationship between the predictor regions and the SCM, and also the influence of internal states and unobserved behaviors on the SCM activities. So we would like to thank you and thank our mentor, Dr. Znamenskiy, and Dr. Muthmann for your instruction and help in this project. We would like to also thank Neuromatch Academy organizers for organizing this event, and we would like to thank you for your attention.